All right, today we are taking a journey into one of the most important places in the entire universe, the nucleus of a cell. I want you to think of it like a microscopic fortress, a high security vault with just one job, protecting the most valuable thing any living organism has, its genetic code. Now get this, inside that tiny microscopic fortress is something that just seems impossible. If you could take the DNA from just one of your cells and stretch it all the way out, it would be over 2 meters long. Yeah, a single strand of genetic code that's taller than most people, all packed inside a structure you literally can't even see. So this brings up a huge biological puzzle, right? How in the world does a 2 meter long molecule cram itself into a space that's completely invisible to the naked eye? Well, the answer is just a total masterpiece of natural engineering. Okay, before we solve that packing problem, let's just cover the basics. The nucleus is the cell's command center. It holds all the genetic blueprints, and its very existence is what defines eukaryotic cells. You know, the complex cells that make up everything from a tiny plant to you and me. So, if we peek inside, we find a few key things. You've got the nucleoplasm, which is kind of like the jelly-like liquid filling the space. Floating in it is the chromatin, which is basically all that DNA, just densely packed. And then there's this other structure called the nucleolus, which is a specialized factory we're definitely going to look at in a minute. To really understand this fortress, we have to start from the outside and work our way in. So let's take a look at the walls, the very thing that protects all that precious genetic material from the chaos happening out in the rest of the cell. These walls are called the nuclear envelope, and it's actually a double membrane. The inner layer is really cool. It's reinforced by this protein network called the nuclear lamina. The best way to picture it is like a dense spider web that provides structural support and gives all that DNA something to hold on to. Of course, a fortress is pretty useless if nothing can get in or out. And the cell's solution for this is just brilliant. It's called the nuclear pore complex. These are these super sophisticated protein structures that act like security gates. They look a bit like a basketball hoop with a net in the middle, and they let small stuff like water just flow right through, but they are incredibly strict about controlling what bigger, more important molecules, like proteins, get to pass. So the security is all set up. Now let's get back to that completely impossible storage problem. 2 meters of DNA in a microscopic space. The way the cell solves this is with an unbelievably elegant packing system. The solution is called chromatin. And it's not just a tangled mess of DNA, not at all. It's a highly organized material where the DNA is wrapped and coiled tightly around proteins. This coiling action creates an insane level of compression, turning that 2-meter-long string into a perfectly manageable little package. But, this compression creates a whole new problem. If your entire library is packed that tightly, how do you find and read the one specific book, or in this case, the one gene, that you actually need? Well, it's no surprise the cell has a really clever system for this too. You see, chromatin actually exists in 2 different forms. First, you've got euchromatin, which is loosely packed. Think of this as the frequently used section of your library. It holds the genes the cell needs to access all the time. And then you have heterochromatin, which is packed super densely. This is like the deep storage archive for genes the cell almost never needs to read. This organization happens on so many different levels. If you zoom all the way out, you see the chromosome, which is the most condensed form. But then you zoom in, and that chromosome is actually a coiled fiber, kind of like an old-school telephone cord. Zoom in again, and the cord itself is made of these things called nucleosomes, which look exactly like beads on a string. And that string? Well, that's the DNA double helix itself. This brings us to the nucleosome, the absolute basic unit of packing. Just picture 8 special proteins, called histones, all glommed together to form a core, like a tiny little spool. The DNA thread then wraps around the spool twice. This whole beads on a string setup is the very first and most important step in the entire compression process. Now, the words we use here can get a little confusing, so let's just break it down. For most of a cell's life, its 46 chromosomes are just loose, kind of like spaghetti. But when it's time for the cell to divide, it first has to copy all of its DNA. Each chromosome then ends up with 2 identical sister chromatids, and they're joined together in that classic X shape we all know. And here's the really tricky part. That X structure, the one with 2 sister chromatids, it's called a chromosome. But after the cell divides and those sisters split apart, the single strand that's left is also called a chromosome. It's the same name for two different things. And that's just something that's really important to keep in your head. Okay, so we have this secure, highly organized genetic library. But how do those blueprints actually get turned into action? Well, for that, we need to go back and visit that little factory we mentioned at the beginning, the nucleolus. The nucleolus is this dense little structure inside the nucleus, and it really only has one job: to build ribosomes. And ribosomes are the cell's protein-making machines. It uses a special kind of DNA to build these crucial little factories, which then get shipped out of the nucleus to go get to work. This three-step process is basically at the heart of all life. It's beautiful. First, a piece of DNA gets copied into a temporary message called messenger RNA, or mRNA. Second, that mRNA message and a brand new ribosome get exported out through those nuclear pores. And finally, out in the main part of the cell, the ribosome grabs that mRNA and reads its instructions to build a very specific protein. And here is one last amazing detail about that whole trip. A fully assembled ribosome is the biggest single thing that can actually pass through a nuclear pore. It's just a stunning piece of evidence for how precisely engineered those security gates are. They're built to let something so huge, yet so essential, get through to do its job. So we've seen the nucleus as a fortress, as a library, and as a factory. It protects, organizes, and copies the actual blueprints for life itself. But that leaves us with one final and pretty profound question. If the nucleus is the library, who's the head librarian? What is it that tells the cell which blueprints to read and when to read them to create everything from a liver cell to a neuron? Well, that is a story for another time.